Hi, everyone. Happy Friday, and welcome to the Myotonic Dystrophy Foundation's Meet the Drug Developer session. I'm Tanya Stevenson, the CEO of the Myotonic Dystrophy Foundation, and today we are going to be talking with Arthex Biotech. Just a little bit about the foundation. We envision a world with treatments and a cure for myotonic dystrophy. Our mission is community, care, and a cure. We support and connect the myotonic dystrophy community. We provide resources and advocate for care. We accelerate research towards treatments and a cure. Just a reminder of the resources and support available from the foundation. We have toolkits and publications, including clinical care guidelines and recommendations at myotonic.org/toolkits-publications. You can learn more about our support programs, including our support groups, at myotonic.org/find-support. Our calendar of all of our events can be found at myotonic.org/calendar/month. And our digital academy, which hosts our presentations from past conferences, all of our webinars that have been held, and eventually in the next week or so, we will be posting today's presentation on the Digital Academy. So that will be myotonic.org/digital-academy. Our Meet the Drug Developers sessions, we have a few more that are coming up. First Friday of the month, 12 o'clock Pacific, just like today. We have one exception. This Wednesday, we will be hearing from Dyne Therapeutics about their top-line data, their preclinical data, sorry. So that will be on a Wednesday, not a Friday. And then you'll hear from AMO and Avidity as the year progresses. We hope that we will see all of you at our conference in Washington, D.C. I would like to introduce who we will be hearing from today, and that is Dr. Beatriz Llamusi. She received her master's degree and PhD in biochemistry from the University Miguel Hernandez in Spain. During her PhD, she focused on the molecular basis of neuronal regeneration. After that, she began working in the Department of Genetics as a postdoctoral researcher, where she dedicated more than 13 years to the research of pathogenesis mechanisms and therapeutic approaches in rare neuromuscular diseases. During this period, she's produced over 22 scientific articles and 7 related patents. She was a PI in 5 competitive projects and supervised 4 PhD projects. During the initial steps of the project that led to Arthex Foundation, she was awarded several valorization grants, including training in business management and development, IPP, financials, and regulatory issues. She co-founded Arthex Biotech in December 2019, has served as the CEO, and currently serves as CSO. We will also be talking with Dr. Judy Walker. She started her career as a clinical neurologist and clinical trial investigator in Canada. She is the Chief Medical Officer of Arthex, and in the late 1990s, she joined the industry and has spent the last 25 years in a variety of clinical and medical leadership roles in the US, Switzerland, the UK, and in Singapore. Her focus throughout this time has been to bring effective and safe drugs to market for patients with some of the most disabling neurological conditions, including many rare diseases. At Arthex, Dr. Walker is responsible for all clinical and medical activities in the company, moving Arthex's lead investigational compound, ATX-01, into clinical studies for myotonic dystrophy, preparing for ongoing development of the drug pipeline, and building a highly skilled team to ensure success. She is excited and humbled by the potential of Arthex's investigational drug, ATX-01, to have a meaningful impact on DM patients' lives. Okay, many of you have sent in your questions for the Arthex team in advance. Thank you so much. Those questions were forwarded to the team. They are going to do their best to integrate responses to those questions into their presentation today. You also have an opportunity to ask questions during the presentation. So if you open the questions tab and you type in your question, you just have to click send, and it will go directly out here to our presenters. So they will have a chance to see that. They will likely be reviewing all of the questions at the end of their presentation. So if you're on a smartphone and you're not on a computer, you can click the question mark icon at the top of the screen, type your question, and click send. If today's presentation speaks to you, please consider donating to MDF. Show your appreciation at myotonic.org/donate. And I would like to now welcome Dr. Llamusi and Dr. Walker. Thank you so much.
Thank you so much, Tanya, for this nice presentation, and thank you everybody for being here today and being interested in what we are Tanya said. I'm Beatriz. I'm really glad to be here with you all today. Arthex is a clinical stage company where we are developing modifiers of microRNAs for the treatment of diseases with high unmet medical needs. Our main product is called ATX01. That is a molecule that we are developing for the treatment of myotonic dystrophy type 1, that, as you know, there are not yet approved therapies for this disease. I will call you later how ATX01 has a role in the therapeutics of the disease, but just to let you know, our main upcoming key value inflection points during the next years will be the IND and CTA submission during this year for the applications for the clinical trials, and hopefully we will have already readouts of these clinical trials in 2024. Yeah, well, this is just to introduce you some of the members of our team. Here you can see the management team, also the scientific advisory board. We are happy to count with really international experts in different areas, mainly clinics and also chemistry. We are here in the Park Scientific in the University of Valencia. We will be welcome you here if you get to Valencia. And if you can see in the next slide, what we are, sorry, it's moving very slow. Yes, what we are developing during the last 3 years in the life of the company is this platform for the rational design of molecules. And what we have focused this and what we wanted to do is really to bring the therapeutic molecules to the tissues that are affected in the disease. So in the myotonic dystrophy, as you know, muscle and nervous system defects are the most prevalent symptoms in the disease. So we wanted to generate molecules that can have a therapeutic effect in these tissues. And in our platform, we are working with 2 elements. One, antisense oligo, which is an anti-microRNA oligo. In this sense, this is an RNA molecule. You should have heard of RNA a lot when all these questions about the COVID vaccine. So RNA became very famous. And what we are doing is to, with this RNA, we are trying to modify the expression of a microRNA in the tissues of the patients. This molecule is a single-stranded RNA with different chemical modifications that we are using to obtain a highly stable and efficacious molecule. But we are modifying this molecule and linking this molecule with a hydrophobic conjugate that is allowing us to target the tissues that are our interest, the tissues affected in the disease. And in addition, it's helping us to reduce potential toxicity of the molecule because it's producing less accumulation of the molecule in kidney and the liver. So finally, with this combination, we generated ATX01. That is the molecule that we are developing into the clinics. We have been developing different, a battery of tox and in vitro and in vivo studies to assess the efficacy of this molecule. Please move to the next slide. Yeah, well, you all know very well, probably better than myself, what is myotonic dystrophy. Although it is considered a rare disease, you know that still a lot of people is suffering from this disease. You can see that the prevalence, the incidence in US based on some screening at birth studies, it's in 1 in 2100. So it's a really very high and undiagnosed and underdiagnosed disease. The symptoms are, although they are very important in the muscle, they are also importantly affecting other tissues. And what we have heard from patients sometimes is that some of the patients that really have a more important impact in their daily task activities are also related to nervous systems, such as daily time sleepiness. So we believe that not only that in our case, one of our main differential factors is that we are not only focusing our therapy in the muscles, because we believe that in this disease specifically, it's important to reach other tissues. Yeah, well, just to explain you the mechanism of the disease at the molecular level, trying to keep it simple, this is the situation in a healthy muscle, where you can see that the DMPK gene is generating the DMPK mRNA, and this mRNA it's producing the normal protein. On the other side, you can see MBNL proteins, which are completely unrelated to DMPK. These proteins are very important regulators of splicing. MBNL is modifying hundreds of other proteins, and in a normal condition, this protein has a role in the nuclei and in the cytoplasm of the cells. But in the DM1 disease that you can see in the next, this DM1 protein normally is inhibiting the production of one microRNA, which is called miR23B. So there is a loop here where MBNL participates in the generation of the mature form of this microRNA, and these two molecular factors needs to be balanced, because otherwise increasing the amount of one of them will affect the amount of the other. And this is exactly what happens in myotonic dystrophy disease, where, as you can see in the next slide, the levels of MBNL are downregulated because this protein is attached to the mutated DMPK transcripts. So as you for sure have heard lots of time, the patients have this expanded or this mutation inside the DMPK gene that is causing a toxic RNA. And this RNA is acting as a sponge of MBNL proteins. As the protein gets sequestered into this ribonuclear foci that you can see in this photograph on the right with these green dots, then there is a lack of function or reduction in the function of this protein, which is causing the aberrant splicing of hundreds of molecules which are regulated by this MBNL. As with this reduction of the MBNL, what we obtain in the muscle is an upregulation of the mature form of miR23B in the cytoplasm. So as you can see, this microRNA has an inhibitory function on MBNL and is suppressing the expression of the protein. So what we have in the DM1 condition, this disease is really very complex at the molecular mechanism of action, because although the mutation is very well described, this mechanism of action is quite complex, and the final result of the sequestration of MBNL in the foci and the upregulation of this microRNA miR23B is that we finally have a loss of function of MBNL. Most of the proteins, most of the companies trying to develop a therapy against myotonic dystrophy disease have been more focused on the toxic DMPK, but we believed that miR23B will be a very good therapeutic target that will allow us to have a higher impact on the symptoms of the patients. If you can see in the next slide, in the next slide you can see how our molecule ATXO1 has an activity on the levels of MBNL, or on the active level. ATXO1 is binding to MIR23B and blocking this microRNA. So it is allowing an overexpression of MBNL. But in addition, this inhibition of MIR23B is destabilizing the toxic DMPK and producing a release of MBNL. So finally, with this ATXO1, what we have, it's a dual effect. So we are not only active on the standard target of the disease, which is the MPK, but we are, in addition, we are active on the general levels of the protein MBNL, which is the one that is more directly linked to the patients to the symptoms of the patients. And finally, this increase of active MBNL is causing this correction of splitheopathy. This is quite unique in the field, and we believe that this will allow us to have a more important rescue of the symptoms. Yeah, this, what we call this dual mechanism of action, or the dual target approach, this effect that ATXO1 has in DMPK and also in MBNL, it's conserved through all these species. So we have seen that every time we use our molecule, we can, as you can see here in the upper part, we can, these are human DM1 myoblasts, or myotubes, sorry, and when we use ATXO1, we have this decrease in the amount of toxic DMPK and also an increase in MBNL. So we have an effect on both, on the genetic origin of the disease and also on the protein that is linked to the symptoms. And the data you see below is a similar result that we obtained in muscle of non-human primates that have been used for the immunotox studies. So we have seen that in the muscle of these monkeys, we also obtain a very similar result, this dual therapeutic approach. And we have also seen this in rats and cells also from mini pigs and in the animals. So it seems that this mechanism of action is quite well conserved. And this is very relevant because it allows us to use exactly the same molecule from the proof of concept in the animal models to the clinical studies in humans. So with our molecule, what we are learning in the mice model, it's really very meaningful to help us to design the best potential clinical trial in the humans. Yeah, as I was telling you, the MIR23B, it's very well conserved, but you see here on the left the conservation at the sequence level. So you can see that in all these species, in humans, mouse, rats, mini pigs, and dogs, and non-human primates, we see this total conservation. So the sequence of the microRNA is completely the same. But in addition, the mechanism of action, as I was telling you, you can see that the results we obtain in cells from rats, mini pigs, and also in the in vivo studies, or in the non-human primates, you can see that every time we are treating with our molecule in these pink bars, we are increasing the levels of our target MBNL, and in fact there is a dose-dependent effect of our drug. Yeah, we have done a lot of work in human DM1 cells because, as you well know, there is a lot of heterogeneity in the mutation found in the patients. Different lengths of the repeats, or there has been also found that there are some interruptions in these repeats that can affect the phenotype. So we believe that it's very important to assess the effect of our drug in different cell lines from patients to really to get to know if we will be able to have an effect in all types of DM1 patients. So doing this, what we have found is that we saw a nice correlation between the dose of ATXO1 that we are using in the cells and the levels of MBNL protein that we are able to upregulate in the cells. So every time we increase the dose of the drug, we are increasing the levels of the protein. In the middle, you can see also that an increase in the levels of the protein is also correlating very nicely with improvement in the splicing of a group of transcripts that are regulated by MBNL. And here we were analyzing some of the splicings that have been shown to correlate very well with strength in the patient. So hopefully this nice rescue of splicing that we can see here will be also observed in the patients. And the graph you see on the right, this is coming from the publications below from Wang, where they have observed that improvement in splicing index has a correlation with the ankle dorsiflexion strength. So this link between a molecular improvement and a functional improvement is crucial because it will allow us to use this splicing index analysis in the tibialis anterioris of the biopsies from the patients in the clinical trial, and this will be used as a surrogate biomarker of efficacy. This is something that you have already seen used by Ionis or Avidity, and the idea behind this, the rationale or the relevance of this biomarker is that it could help to make a faster development in the clinics if we see this early improvement with this biomarker. We have done a lot of studies in vivo using efficacy studies using HSALR. This is the mice model, which is the best one considered for an adult onset DM1 model, because these mice are expressing around 220 CTG repeats in the muscle. So they have only the toxic repeats in the skeletal muscle. Here we were doing what we will be doing in the patients, which is one intravenous injection every 2 weeks, and in total 3 injections. And then we sacrifice the animals 2 weeks after the last injection to make the assessment in this case specifically. But just to let you know, we have also done some studies at different time points after the last injection, and what we have seen is that the effect is endurable at least until 10 weeks after the last injection. So we believe that we will be able in the future to give a more comfortable dose regime or administration regime to the patients. But here in the first study, we will be doing this bi-weekly administration just to make sure that we can reach proof of concept in the first study. In these animals, what we can observe is that we could rescue, we were treating with different concentrations of the compound from 6 mg/kg to 24. And just after the first injection, as you can see, the strength of this animal was already increasing until the what will be considered the control condition here in this purple rectangle. And in addition to this functional rescue, we could also observe a molecular rescue. We could observe the PSR, means percentage of splicing rescue. Every red dot that you see here is a composite of 5 splicings that have been quite frequently analyzed in these mice and are also very well conserved in the human patients. And as you can see, there is a nice dose-dependent rescue of these splicings. And most importantly, even at a very high, a very low dose here at 6 mg/kg, we already observed more than 20% of splicing rescue. And this is relevant because the studies performed by Wang in a cohort of patients have shown that 20% of rescue of splicing was already functionally relevant, because this could be the difference in 1 grade in the ankle dorsiflexion strength test, which might be the difference between walking and non-walking in some of the patients. So there is no need to have a high percent, 100% of rescue of the splicings to already have an important impact on the symptoms. In the graphs on the right, what you can see is what we call the target engagement. So how is the impact that we have on the MBNL protein levels? This is a quantification of MBNL upregulation. in different tissues of these mice. And what you can see is that in the muscles, heart, diaphragm, and also in the brain, there is a nice dose-response upregulation of this protein. This is very relevant because this means that we could be treating all these tissues. And especially the brain is especially difficult to reach using intravenous injections. And as I said, we are quite proud that we will be able to help to improve also symptoms in the brain. And what is more important is that when we go to other organs, such as the liver and the kidney, we only have target engagement, or we only have a slight upregulation of the protein at the highest concentration. So that means that we have a preferential MBNL upregulation only in the tissues affected in the disease. And although liver and kidney are the main important tissues involved in the clearance of this molecule, we are not having such a strong effect on these tissues. Please move to the next one. Yeah, we have done in the animals the similar thing as we showed you previously in the cells. What we have done is to study the correlation between the dose of ATX that we were giving and the protein. And also, we saw a correlation between the levels of the protein that we study in the muscles and the splicing rescue. So you can see that with higher levels of protein, we obtain higher rescue of splicings, this group of splicings here specifically. And finally, we have seen a nice correlation between a molecular effect, which is the overall percentage of splicing rescue, and a functional effect, which is the rescue of strength. So this correlation will help. Will be very helpful in the clinics if we observe something similar in the patients. With this molecule, with ATX01, we have already completed all the toxicology studies required by the agencies in order to submit the first-in-human study in patients. The results we have obtained is that ATX01 is remarkably safe and well tolerated. It did not induce genotoxicity. It was not immunotoxic in non-human primates. And the therapeutic window that we have observed is wide enough to support ascending doses in the treatment in humans. In addition, the starting dose that we are planning to use in humans is the equivalent to the therapeutic dose that we have observed in the mice model, where we already see a nice effect or therapeutic effect of the drug in the muscles. Well, we are just completing this preclinical package. We already have all the results, and we are just working on the dossier with the idea of presenting the IND submission to the FDA at the end of the summer. I think that now comes already the clinical part, so please, Judy.
Thank you very much, Beatriz. That set the stage very nicely for what we're doing in the clinical study. So as Beatriz mentioned, we are getting ready to start our first-in-human study in patients with DM1. This is going to be a phase 1/2A first-in-human study. It's going to be double-blind, so the patients who are participating won't know if they're on active drug or placebo, nor will the doctors treating them, and nor will us, the team behind that's managing the study, until the very end of the study when we reveal who got what. We are doing a single and multiple ascending dose study, so very similar to some of the designs I think you've seen before. And we're targeting classic myotonic dystrophy type 1, i.e. the adult form of the disease. We'll come back to the different types of DM and where we think this drug might work. But for the first study, we're being quite focused on adult DM1. So the goals of the study, our primary goal, like with every first-in-human study, is to make sure our drug is safe and tolerated in patients when we give it as a single dose or when we give it as multiple doses. Excuse me, I've got a bit of a cold, so apologies. As Beatriz mentioned, our toxicity program was really, really good. And because we are using the exact same molecule in humans that we used in animals, there's no sort of slight changes, that we're fairly confident, but you never know. And again, this is why we have to be very cautious when we do a first in human study and only study a small number of patients, and be very cautious in how we're starting with a low dose and slowly increasing the doses. Aside from safety and tolerability that we're going to assess, we also want to look at target engagement, i.e., does the drug get to the tissues it needs to get to, in particular the muscle, and does it interact in the muscle the way we think it should and do the things it should? So such as increasing the MBNL levels and improving the splicing index. So we're going to be doing muscle biopsies. Yes, I know they're not the most fun, but it's the best way to tell if we're having an effect. So we're going to do muscle biopsies and look at the MBNL levels and the splicing index. And then we're also going to look at clinical endpoints. So we're going to look at the muscle measures, the things that you're familiar with, the VHOT and walk-run tests and so forth. But because, as Beatriz pointed out, our drug isn't limited to action in the muscle, we believe it could have action elsewhere. We're also going to start to look at some other tissues and to see if we can show any effects there. And of course, what's going to be really important, it doesn't necessarily matter that you can improve, muscle strength by a minuscule amount. What really matters is how does that affect the patient's ability to do things? How does it affect you? And so we're going to look at a number of patient-reported outcomes to see, is it actually having an impact on your lives? So the design of the study, I said there's two parts. There's the SAD part, single ascending dose, MAD, multiple ascending dose. So in the SAD, patients will receive a single dose. We're going to have 3 cohorts. So as the cohort 1 is a low dose, but as Beatriz pointed out, still active in the mouse, the equivalent dose in the mouse, not the same, but the equivalent dose in the mouse. And then we're going to have another cohort that's going to be of patients who are going to get a higher dose, and then we're going to go up to a 3rd dose. We might possibly go higher if we need to, but we're planning on doing at least 3 cohorts. So what the cohort means is that there will be 8 patients in each of these cohorts. And within these 8 patients, 6 will be on active treatment, 2 will be on placebo. And as I said, nobody will know who's on what. But this will give us an idea of what are the effects after a single IV dose. And the drug is given IV. And we will follow the participants in the study up for 12 weeks after that dose. Now, in the MAD part, it's the same idea. We're going to have 3 cohorts. We're going to look at the same dose levels, starting with a low dose and then increasing again. In each cohort, we're going to have 6 active and 2 placebo patients. But the only difference is that instead of getting a single dose, patients in the MAD will get 3 doses, 2 weeks apart, just like we did in the HSA-LR mouse model. So 3 IV doses 2 weeks apart. And then after the 3rd dose, we follow the participants up for another 12 weeks. And then that will be followed by a long-term extension study where we can do longer-term follow-up and provide drug to all patients that are, so long as the results of the main study show that it is safe to do so. The study we've called the antimiR study. Obviously, concatenation of our name, Arthex, and MIR, microRNA. So it's the antimiR trial. And we're hoping to get this going soon. So the big question we always get, and I've also seen it in the pre-questions, is who can enroll in this trial? Well, I said we were looking at adult DM1. So we're looking at adults, male or female, ages 18 to 64. But not only is there age 18 to 64, we don't want patients who are congenital DM1 who've grown up. We really are focusing on the adult type for this study. We want patients who have a diagnosis of DM1 confirmed by more than 150 CTG repeats. And we're looking for patients who are relatively early in the disease course, who are ambulatory. They can still walk. We want patients who have myotonia, and we're going to measure that with the famous video hand opening test, and we want a VHOT score of more than 3 seconds. And obviously, we want the patients to be well informed about what they're getting into, and they have to sign informed consent. Now, who is not eligible for this first study are the patients with congenital DM. Anybody who has a contraindication to muscle biopsies, either because your blood doesn't coagulate, doesn't clot well, or for whatever reason. Patients who are on anti-myotonic medication, if you've been off the anti-myotonic medication for a few weeks, you can participate, but we don't want you on it in the study. And we're also being very careful about patients who have a moderate to advanced cardiac disease. There are a number of patients who unfortunately will be excluded from this first study. And it's because the first goal is to make sure patients stay safe. If we see, and we have not seen in any of our animal work that there was any cardiac problems induced by our drug, but we know that the DM1 population, some people will have cardiac disease, and we don't want to take any chances. So as you can tell from all these criteria, we're aiming for a population that has DM1 but is relatively healthy otherwise. And I think that's the best way for a first in human study. Later, as we move, as we gain confidence with this drug and we see, hopefully that there isn't any major safety issue, we can broaden to include patients who have more complications, who have more advanced cardiac disease, who are not ambulatory. But for the first study, I think we need to be very, very cautious. So, and what are we going to measure? So those are the patients we're going to put into the trial. I've told you what we're going to do in the trial. We're going to give the drug either a single dose or 3 doses. And then what are we going to measure? So this is what we call our outcome measures. And the first thing, of course, is safety and tolerability. So we're going to ask you about any adverse events you've had, anything unusual that's happened. We're going to do some blood work and take some urine samples and test that for various things. We're also going to do some electrocardiograms, and we're going to do what's called a Holter test, which is a 24-hour portable electrocardiogram. We're also going to measure the level of the drug in your blood and urine at different time points. And that's called pharmacokinetics. And we're going to look at what we call pharmacodynamics, which is how does the drug behave in the tissue, and in this case, the muscle tissue, which means, as I said, muscle biopsies. And there we're going to look at measuring the MBNL protein levels, which should go up in the patients treated. And if it replicates what we saw in the animal, the higher doses will have more MBNL. And we will look at the splicing index, which should improve, and that should improve also in a dose-dependent manner if it's the same as we see in the animal. But we're also going to look at a number of different clinical efficacy measures, because it's nice to have high MBNL levels, but what's really important to you, I'm sure, is being able to do things. So we're going to do a number of different muscle and function testing. So some of these I'm sure you've heard about. We talked about the VHOT, 10-meter walk-run test, timed up and go, time sit to supine, quantitative myometry, and these are all fairly standard muscle tests that I'm sure many of you have already had. We're going to measure your breathing with the forced vital capacity, and we have a few other tests that we're going to add as well. But those are tests that directly measure how well your muscles are working. We're also going to ask the treating doctor to make some evaluations. So we're looking at because we think our drug might be active in the central nervous system, we're going to look in the brain. We're going to look at an apathy evaluation scale. We're also going to ask the clinician for his or her impression of how things have changed. And then, of course, what's really important is the patient-reported outcomes. At the end of the day, it doesn't matter if your 10-meter walk-run test improves by 3 seconds. What really matters is how are you doing? And again, because of the brain, because we think our drug might get into the brain and treat some of the brain aspects, we're going to look at a fatigue and daytime sleepiness scale. And we're going to look at the DM1 active scale, which is a patient-reported scale of how they're doing. We are also going to look at the impact of the drug on different on your activities of daily living, your ADLs, but also of particular symptoms on your ADLs. And one of the things we're doing as well is, one of the things we've heard from the community is that GI symptoms can be really bothersome. So we want to know, are we having an effect on those? Are we improving the GI symptoms? We don't know. We don't know if it gets into the gut, but it's fairly widely distributed. So it's something that we're going to check in this study. So when and where are we doing this study? So as Beatriz said at the very beginning, we're hoping to file an IND. So filing an IND in the US is the first step. You have to file an IND before you're allowed to start a study. So we're going to file an IND in Q3. Currently, that's looking like around September time frame. And we're going to do the similar procedure in the other regions after that. And the reason there's a difference between the US is because the different regulators in different countries want to see different things. So we need a little bit more time to gather the information for the other countries. So, but the US will go first. And then we expect to have our first patient in the fourth quarter of 2023. So I'm hoping we get the first patient in by December 2023. So that's this year. And then we should have early results from the SAD even by late 2024. So that can come pretty quickly. Now, where are we doing the trial? We're going to be doing it in the United States. We are in the process of trying to determine which centers we'll be doing it. We're trying to have a geographic distribution, so it's not all in California and nothing anywhere else. So we are trying to get a few across the United States. We have one site in Canada that we think will be a good place to start in Quebec. And we have a few sites in Europe that we're looking at right now, Spain, Netherlands, Italy in particular, and possibly the UK. So we're sort of considering coming to the UK as well. So those are the sites. We're working through an organization called the Myotonic Dystrophy Clinical Research Network. And as you know, this network is growing day by day, and we might be able to add more sites, but at the moment, these are the sites that we're going to be working with. So that's our first in human study in adult classic DM1. And this shows you our pipeline. So this drug, ATX01, we believe could have an impact in DM1, but we also think it could have an impact in DM2. And the reason is because we're acting on the microRNA, miR23B, which increases MBNL. We know that MBNL or lack of MBNL is also an issue in DM2. So even though the gene itself is different, because our drug doesn't directly target, it's not specific to the DMPK gene, it has the potential to work also in DM2. Now, there is no animal model for DM2, so we're looking at some cell lines from patients who've with DM2 to look if we have an effect there. But that's something that would be next on our list to look at. And of course, I mentioned that we started with DM1, adult DM1, but we will also have a plan to look at congenital DM1 and look at the younger patient population, because this is clearly an important population. And there's no reason to think that we wouldn't work in the congenital DM1 population. But it's always wise to start with an adult population before going to a younger population. The thing that Beatriz pointed out is that we have done these studies in cell lines of patients who have varying lengths of CTG repeats. So, and we've shown that even if somebody has very few CTG repeats or very long repeat lengths, we have efficacy. So again, that sort of supports the fact that we might be useful in the congenital DM1 population where the repeat lengths are longer. And we are working on some other drugs which probably don't interest you at the moment, but this is where we're at now, and we're very, very excited. And I think if we see nice effects in our first study, we could kick off programs in these other conditions. So I think I think this is where we wanted to stop, and I hope we've got across the message that, and not just from us, from this series of talks, that there's a new age dawning in the development of agents for myotonic dystrophy. We are truly excited, and we're very grateful to be part of this. This is a very exciting time, and you are the community of DM1 patients are so important to us. Excuse me. And we talk about, and we have to be, as scientists, we have to be very careful. We don't want to overpromise. And I know that many of you have asked questions, is this just going to relieve the symptoms, or is it going to cure the disease? And the truthful answer is we don't know. But if you take what we've been telling you, it looks like this should be more than just relief of symptoms. It will relieve the symptoms, but if it does in the human what it does in the animal, we should see some reversal of disease. I wouldn't, cure is a big word, but we should see some reversal of disease. So I think this is extremely exciting. We won't know until we do the clinical studies, but I do believe there is reason for hope. And on that note, I will pause and remind you that if you want to ask questions, as Tanya said, go to the questions tab.
Yeah, we can maybe go to the. Sorry, I have the list of questions here that were sent in advance, and I think most of them were already replied during the presentations, but I have some of them, Judy, that I think we have not addressed, so maybe I can go through them. Sure. One of them was, can multiple people in one family join the trial?
Yeah, I think if multiple people in one family are eligible, I don't see a problem.
Okay. Let me check. Participant time. We already speak about DM2 congenital. Yeah, one of them were questioning if there is any plan to carry out the clinical trial in Spain.
That is our hope, indeed. Yes, we've been in conversation with a very well-known specialist in DM1 in Spain, Dr. Adolfo López de Munain. We visited his site. He's very keen, so fingers crossed that will work out.
Okay. Another question was, will this be a good psychiatric treatment for social ineptitude?
That's a very good question. I don't know. But what I can tell you is that in animal, the drug gets into the brain and we see what we call target engagement. In other words, it increases the MBNL. So it's doing something. It's not only getting into the brain, it's getting to where it needs to be, and it is doing what it's supposed to do, i.e. increasing MBNL. So I think that's the first step. It will be, we'll have to wait and see. I mean, it's certainly something that we're very interested in looking at. I think there's a possibility, but it's hard to know at this stage. Fingers crossed.
Mm-hmm. Now there is another, this is a good one also. When will we know the final results of ATX01 trial in Europe and in US? And also, if everything goes well, when will it be possible to start the treatment in patients, or when will it be approved?
So it's a multifaceted question. Again, a very good question. So we would expect to get the results of the study around in the 2025 timeframe. We'll get earlier results sooner, but to get the final results 2025. Now, doing this one study isn't enough to get a drug approved and for it to be available on the market. So what will need to happen is that if we get good results from this study, we may be able to go straight to a confirmatory study. And this would be a much larger study, a broader study that would assess the effects in a greater number of patients. And then that would probably take a few years. And then it would go through to the regulatory agencies like the FDA in the US and the various other countries have similar regulatory agencies. And they would determine whether the balance of risk-benefit was sufficient to approve the drug. So it's a long haul. However, one of the things that we will consider doing is offering an expanded access program. And sometimes that's a way, if there's a treatment that really is showing benefit, we can make it available. It depends on the country, so I don't want to overpromise. It depends on the country, but sometimes early access or expanded access programs can be very helpful to get drugs that could be beneficial to individual patients, even if it's not approved by the regulatory agencies.
Okay, another very good question. Any noted side effects? Not in the animals. As I said, we have already completed all the previous studies required by the agencies in rats, mini pigs, and non-human primates. It was shown to be very safe, in fact. Even at the highest concentration we tested, we have not seen toxicity. In fact, in the chronic studies, in the animals that we will be performing in parallel to the clinical study in the patients, we are going to go even to higher concentrations, just looking for tox effects, because we have not seen any. And in any case, as Judy mentioned, we are going to be very careful in the clinical trial. Okay, let me see. There was another that I liked a lot also. Effects. Let me see. I cannot find it. Yes, how is your approach different from the other trials in progress?
I think what's different about our study relates a lot to the compound itself, and because we have a dual mechanism of action, because we have shown target engagement in different tissues, we are going to be studying those other tissues and looking whether we can see early signs of effects. The study design isn't hugely different from what other people are doing. We are being very cautious, and it's just not because we think there's any issue with our drug, but because you just never know. And we know we just never know what can happen in a clinical study. So we are being very cautious with how what type of patients we're enrolling in the study, but our study design isn't that different. I think it's more that the molecule itself is very different, and its mechanism of action is very different. So it could be very interesting to see how it does compare to other treatments.
Because the ones
Would you tell us what an IND is?
Yeah.
Okay, that's a good question. So an IND stands for investigational new drug, and in the US, it means that you are allowed to run your clinical study. So when it's the first time a drug is tested in the US, you have to go to the FDA, which regulates drugs in the US, and ask them, can we do this study? And they require a lot of information. They require a whole dossier. They want to know about your product, how you made it, how it's synthesized, what its composition is. They want you to show that it's stable. They want you to show that it's free of toxins. They want you to show that you've tested it thoroughly in animals and you've done all the right tests. And they want to know what your study is, and they want to see what you're planning to do with patients. And then if they say, okay, this seems sensible, they will let you go ahead and do the study. And that's what we talk about, an open IND. So we submit our plan, and if we don't hear anything back in 30 days, it's approved, unofficially approved. We can go ahead and start our clinical trial. But more often than not, when the FDA starts looking at the file that you submit, then they'll ask you questions, and then you answer them, and you try to get to the point where you get resolution of the issues, and then you can go ahead and start your study. So you may have heard about some programs being put on clinical hold, and some of them are put on clinical hold when the drug is already in a clinical trial. But there's been a number recently in neuromuscular diseases, in DM1, that have been put on hold before the IND is even opened, and that can happen. But we need to in order to do the trial in the US, you need an open IND. The process is a little bit different in other countries. It's similar, but in every country you have to get an approval from a regulatory agency. And in other countries, they call the process a CTA, a clinical trial application. You also, aside from the regulatory process, each center where the study is being conducted will have an ethics committee or an institutional review board that will review the study and review also the data package and make sure that it is safe and ethical and appropriate to be dosing humans. So there are a number of different we need to be sure. We don't do, drug companies don't do studies willy-nilly. We have to be sure that and we have to prove to others that what we're doing makes sense, that we have control of the risks, that we know what to do in case an issue happens. And that's what we need to show the regulators. And the process by which we do that is the IND. So you can't start a first study without an open IND. Subsequent studies, you don't reopen an IND, you just add studies to your open IND. In Europe, it's a little bit different, but in the US, that's how it works. You open the IND for a drug and an indication once. There's another question here. I joined late, so I'm a bit confused. I thought we want to minimize the amount of it in the muscle as it is a signal that the protein, but it sounds like you want a higher level MB now. Yeah, so I'll let Beatriz answer that, but yes, we do want a higher level of MBNL. So low MBNL is what is causing a lot of dysregulation of splicing. And to regulate the splicing, you need to have the MBNL. So you need to increase it. Mm-hmm. It's the last question. Oh, okay. So there's another question here. To understand this, so that accumulates in damaged muscle and can be visually by seriously. Yeah, so yes, the MBNL protein accumulates in the hairpin loops of the toxic mRNA, the MPK mRNA, and that you can see, yes, but because it's accumulated in the hairpins, it's not available in the parts of the cell that it needs to be to do its job. So the issue is that the MBNL hasn't gone away in DM1, but it's stuck. It's as Beatriz said, there's a sponge there, the toxic DMPK that is sponging up all the MBNL, so it's not available. It's as if it weren't there. So what we do is we increase the MBNL, and that makes more MBNL available, and we release it. So we have increased production, release, more MBNL available. That regulates the splicing and hopefully improves the symptoms. And then this one doesn't look like you have DM, but one cannot distinguish DM1. So you can't distinguish DM. Well, in both conditions, you're right. There is increased MBNL, and there's sequestration of MBNL in the toxic RNA foci in both DM1 and DM2. That's same, but the clinical presentations are very different from DM1 and DM2, and there's other ways. The genetic defect is different, so there's other ways to differentiate them. But because the issue in both of these is lack of MBNL, and because we directly increase lack of MBNL in the areas where it needs to be, and because we increase the MBNL, we could have an effect in both. And then what do we have to do if we want to be part of the trial? Well, I think the first thing to do is to speak to your neurologist and discuss with him or her about your interest. As I said, we have a limited number of sites that we're going to be working with. And the question is then to get in touch with the clinical trial center that is running the study to see if they might have spot for you to be screened. So the first step is that we have to screen. There's a lot of patients with DM1, and not everybody's going to meet those inclusion-exclusion criteria we showed you. But we will ask the doctors in the different centers to screen and find the right patients for the study. We will also be posting our study on clinicaltrials.gov, and in there you'll see a list of the clinical trial sites that will be participating, and you can contact them directly as well.
I think, Judy, also there is some questions about the biopsy, where it is done, the biopsy, and also if it's painful.
Yeah, so biopsies, I mean, I'm not going to pretend that they're fun. The biopsies are going to be done in the tibialis anterior, which is like your shin muscle. They're going to be done as a needle biopsy. So it's, you're not going to get an anesthetic and have to do an open biopsy. We're going to try to minimize the number of biopsies that we have to do. So each patient will get, at a minimum, 2 biopsies, one before and one after treatment. If you're we're allowing patients to go from the single ascending dose study to the multiple ascending dose. So if you're in both of those parts, you might get 4 biopsies, but at least 2 if you participate only in one part. When would the trial start in Canada? That's a good question. Probably around the same time as Europe, I'm guessing at this point in time. So we're talking about probably early 2024, sorry, apologies, 2024. What is this? What time are you thinking? What timeline are you thinking? So I think what we need for DM2, I think we need to see something encouraging from DM1, because it's we don't have an animal model, so we need to have something that would give us some encouragement that it could work. And I think if we see a result in DM1, even if we see a result in the first cohort, we could say, oh, well, look, there's something here. Let's maybe look at DM2. Is the compound based on an injection or pills? It's an injection. We talked about muscle biopsy. Is muscle biopsy painful? I'm not going to lie to you and say it's not painful. It is painful, but it's a short-lived temporary pain. I know. I mean, I think Tanya probably would have more feedback from the community about that. I think it's sort of a necessary evil. We don't really know what's going on unless we do the muscle biopsy, to be fair. Although, and I think, you know, and the reason is because we have these biomarkers that are in the muscle. The MDNL level and the splicing defect. I would love to see biomarkers that don't require muscle biopsy, but where the science isn't there yet. I would love to see a blood-based biomarker where you just do a blood test. So, no centers in New York City. I don't think we are looking at New York City, but you're not too far from other centers that we are looking at.
There was, in fact, one question from Tanya specifically about this. If someone doesn't live near one of the trial sites, will there be resources available to help participants travel to trial sites?
Yeah, we are looking at that, definitely, because we want the right patients, and we want to be able to make sure that we remove any barriers for patients who are truly good candidates for the study to being in the study, definitely.
And finally, thank you, Julie, for the congratulations about how we explain something very complicated, because it is really very complicated. And sometimes the scientist doesn't realize that some of the things we say, we are making things sometimes overcomplicated. But in this case, this disease, it has a very complex mechanism with a lot of different molecules involved. So it's, but we really know that the patients are the real experts on the field. I have talked to some patients that could, that knew even more details than myself about all the mechanism of action. So we are sure that you understand, and if not, you will ask. So thank you for your questions, and well, thank you all for your attention today. We were very glad to have this opportunity.
Thank you so much, both of you. Thank you for sharing with us. It is really hopeful, and I agree. You absolutely did a fantastic job explaining something that is usually very complicated for folks to comprehend. We're excited about the possibilities. We're waiting for an announcement of the trial very soon. I do want to share that there is a DM2 mouse model in development, so I want to reassure folks that is happening. There are multiple sites actually working on that, and they're making really good progress. Hopefully we'll actually be hearing about some of that at our conference in September. So please sort of, tune in if you have the opportunity. We would love to see everybody there. Thanks to everyone who joined today. Just a reminder that this presentation will go on to our Digital Academy in the next few days. Judy, thank you for sticking with this. I appreciate it, particularly when you have a cold. You still are here for the DM community, and we are grateful to both of you for making this happen.
Thank you. It's just a cold.
Thanks to you for organizing.
Thank you.
We look forward to hearing more about your progress at the conference in September, and we look forward to seeing everybody there. So thank you again, and have a fantastic weekend.
Yeah,
Thank you. Thank you, everyone.
Bye-bye. Ciao.
Bye.